Hi everyone, we are from Group 19 and my name is Taning Singh. In this video, me and my group members Anne and Jane will explore the key issues of drug testing and validation related to animal use, highlight the 3R strategies, and consider the alternative approach that support more ethical and effective research. Do you know what is drug testing and validation? It is important in clinical research, often involve animal models to assess safety and efficacy. However, ethical concerns and scientific limitations have led to the adoption of 3R principles, which are replacement, reduction, and refinement. This is to minimize the animal use and suffering. In genetic engineering studies across fields like oncology, neurology, and infectious diseases, applying this principle is particularly important.
Hello, I'll be explaining the replacement strategies used in drug testing and validation, which follow the replacement principle of the 3RS. These strategies aim to reduce the use of animal by using alternative methods that can still give reliable results, sometimes even better than animal testing. First, organ-on-a-chip technology uses small lab-made devices that stimulate organ humans, like the liver or heart. These chips allow researchers to observe drug responses in real time without testing on animals. Second, 3D human cell cultures, which involve growing human cells in three-dimensional forms that behave more like real tissues. These provide more accurate results compared to flat cells or animal models. Third, in silico models or computer simulation are used to predict how drugs behave in the human body. This model uses artificial intelligence to support early screening and reduce the need for animal use. There are some examples of a case study by Zhang et al. 2019 show that a liver-on-a-chip device could successfully mimic liver damage caused by acetaminophen. These results were similar to those seen in humans, proving that this technology can replace some types of animal testing. These replacement strategies help improve research ethics, reduce animal use, and provide better data for human drug safety.
Next R represents reduction that aims to minimize the number of animals used in scientific experiment while obtaining a reliable and valid result at the same time. Reduction can be achieved by improving experimental design and the usage of advanced technologies such as the usage of biomarkers and microdosing in humans. Biomarkers are those measurable characteristics such as the concentrations of protein, metabolites, and chemicals found in the human body, which can indicate the drug efficacy. As the animals have different body structure compared to humans, this causes the drug testing to be lacking in accuracy and requires more animal models for validations. By observing the biomarkers in humans, drug responses and the toxicity can be predicted before large-scale animal testing. Biomarkers can also speed up the study duration as the drug effects can be assessed very quickly as compared to animal models. The period needed for drug development can be shortened and potentially hasten the treatment process for the patients. Next, reduction of the animal models for drug testing can be achieved through microdosing. By microdosing in humans at first, which is the phase 0 trial, the dosage of drug given to human volunteers are controlled. This ensures that the side effect of drugs can be reduced to minimal and the drug effects are more accurate when it is used on humans. Furthermore, the researchers can still study the metabolism of drugs regardless of their low concentration. If the metabolism of the drug is low, the drugs can be dropped and reduce the need for further animal testing.
Refinement in drug testing aims to reduce animal suffering by improving experimental methods. In toxicology studies, one key refinement is the use of early human endpoints, which allow the researcher to stop experiments before the animal experiences severe pain or distress. These endpoints are based on early signs such as weight loss, behavior changes, or biomarkers, ensuring timely intervention or euthanasia. Talking about its alternative approach, advanced technologies like AI and machine learning offer powerful alternatives to reduce reliance on animals. These tools can predict drug toxicity and interaction using large datasets, identify potential adverse effects before in vivo testing is needed, and also help design safer compounds early in development. By combining human endpoints with predictive technologies, researchers can both protect animal welfare and enhance the efficiency of drug testing.
To conclude, drug testing and validation are vital steps in the development of safe and effective treatments. However, the ethical use of animals in this process demand careful consideration. The 3 R's principles offer a responsible framework for improving animal welfare while maintaining scientific integrity. By adopting alternative methods, minimizing the number of animal use, and refining experiment procedures, researchers can advance medical progress more ethically and sustainably as technology continues to evolve. Integrating these principles is not just a regulatory requirement but a scientific necessity for future-ready clinical research. Thank you, and that's all from our group.